Glycolysis, breaking down of glucose to pyruvate. So this is the metabolic process which happen in the presence of oxygen. This pathway is otherwise called EMP pathway. It is named after the scientist who worked on it, Emden's, Mayrhof and Parnas pathway. It has two phases, preparatory phase and the payoff phase. In the preparatory phase, ATP is utilized and in the payoff phase, ATP is gained. The glucose is a 6-carbon sugar which is converted to 2 molecules of 3-carbon pyruvate. In the first phase, preparatory phase, glucose 6-carbon sugar is converted to glucose 6-phosphate in the presence of enzyme hexokinase. Hexo 6 kinase, it having the function the kinase which means addition of phosphate. So the enzyme's function is to add phosphate to the corresponding position. So the 6th carbon is the position where the kinase is going to add phosphate. That comes the name glucose 6-phosphate. Here the first ATP molecule is utilized. ATP is the high energy molecule adenosine triphosphate which is converted to adenosine diphosphate. 3 molecules of phosphate will be utilized by the glucose. The 1 phosphate molecule is added to glucose. The second step is conversion of glucose 6-phosphate to fructose 6-phosphate in the presence of enzyme phosphohexose isomerase. Isomerization is changing the structure. These glucose 6-phosphate and fructose 6-phosphate are having the same chemical formula but having different structure. That comes the reaction. It can be able to reverse itself at any point of time. The third step is the second priming reaction. Fructose 6-phosphate is converted to fructose 1,6-bisphosphate. At this stage, the second ATP molecule is utilized. The phosphate which comes out from the ATP is added to the first carbon position of the sugar. That described by the enzyme name phosphofructokinase 1. Kinase which having the function to add phosphate at the position of the first carbon of the sugar, that is to the fructose. That comes the name fructose 1,6-bisphosphate. Fourth step is the cleavage reaction. There is 6-carbon sugar is converted to 2 molecules of 3-carbon sugar. That is in the presence of enzyme aldolase. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are converted from the fructose 1,6-bisphosphate. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphates are 3-carbon sugars. In the presence of enzyme triose phosphate isomerase, the dihydroxyacetone phosphate can convert it to glyceraldehyde 3-phosphate. There comes 2 molecules of glyceraldehyde 3-phosphate. At this point, it will enter into payoff phase. Glyceraldehyde 3-phosphate, that is 2 molecules of glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. Here NAD, that is nicotinamide adenine dinucleotide, is having a low energy which is converted to high energy molecule 2-NADH. Here there is an addition of 2 phosphate molecule. So here there is an oxidation reaction and phosphorylation reaction. The glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate in the presence of enzyme glyceraldehyde 3-phosphate dehydrogenase. Seventh step is 1,3-bisphosphoglycerate, that is 2 molecule, is converted to 3-phosphoglycerate in the presence of enzyme phosphoglycerate kinase. Here, this is the first step, the ATP formation happens. The phosphorylation happen, 2 molecules of ADP, that is adenosine diphosphate, is converted to high energy molecule ATP. Eighth step is conversion of 3-phosphoglycerate to 2-phosphoglycerate in the presence of enzyme phosphoglycerate mutase. Ninth step is 2-phosphoglycerate is converted to phosphoenol pyruvate in the presence of enzyme enolase. The 10th step is the final step. Here also, at the second time, we are gaining ATP molecule. Phosphoenol pyruvate is converted to pyruvate in the presence of enzyme pyruvate kinase. Here, 2 molecules of ADP is converted to ATP. In this payoff phase, there is a gaining of 4 ATP molecule plus 2 NADH molecule. So, totally we gained 4 molecules of ATP plus 2 molecules of NADH. The net gain of the glycolysis is 4 molecules of ATP plus 2 molecules of NADH minus 2 molecules of ATP. That is 2 molecules and 2 molecules in the preparatory phase is subtracted to the payoff phase. So, net gain is 2 molecules of ATP plus 2 molecules of NADH. This is all about glycolysis.